Why others are not joining? I don't know. All right. Um, so good morning, and we will continue our journey on the gene cloning part. This is the concluding session on gene cloning, and protein purification. From next class onwards, we will look at therapeutic cloning, the other forms of cloning. Okay, and I would like to, give an overview and then maybe a couple of classes on the applications of those things. Okay. So in our last class, we actually saw, after you express your protein in E. coli or in, whatever expression system you decide to choose, could be yeast, could be insect cell, could be mammalian cell, right? So I again keep on emphasizing this fact that when you clone a gene and when you try to express a gene, you will not know to begin with which system will work for you. So people generally start with expressing it in E. coli, and then they will eventually, they will decide whether it is working or not. How will you know whether your this thing is working or not? For example, let's say you are trying to purify, say, an enzyme, say something like topoisomerase or DNA polymerase. Taq polymerase, by the way, is now expressed in E. coli, right? So suppose you want to know whether the recombinant Taq polymerase that you expressed in E. coli, is it working or not, you need to have an assay. You need to have an enzymatic assay. And once you know that this enzymatic assay works well and the purified protein from E. coli is working as per the expectations, you will start proceeding with the E. coli expression system. Okay. If you realize that you have purified a protein, but the protein is dead means it's no longer active, then you are going to have an issue, right? So you don't want to be dealing with those kind of situation. Then you will try to express it in, move from E. coli into yeast or into baculovirus or mammalian system in general, okay. When you try to express proteins from, say, some other prokaryotic organism, for example, as I said, gave an example of Taq polymerase, E. coli is fairly a good system, okay. And by and large, it also works for many of the therapeutic proteins, okay, like the recombinant insulin that I've been quoting works very well in E. coli, okay. And by and large, when you try to do this mammalian expression systems, you will realize that anything that has extensive post-translational modification, the ground rule is that it may work, it may not work. So if it doesn't work, we are not surprised. If it works, we are not surprised either. Okay. And then once we realize that the mammalian protein is, depending on your enzymatic assay, if it works, then we will go ahead with the E. coli system. If it doesn't work, then we will generally, people used to use yeast. But I think yeast is, not being used as popularly as it used to be used earlier. Even today, yeast is a expression system of choice. Typically, the hepatitis vaccines that you receive are from yeast. Okay, yeast surface expresses the hepatitis protein. Okay, so it is not that it is abandoned, but many people in the academics have shifted to insect cell and to mammalian cells. Okay. And whether it is yeast or insect or mammalian cell, as long as you're doing it for your academic activities, like, trying to understand the protein function, trying to understand the structure and the activity relationship, you are okay. You have no issues. But when you want to do it for production, that means for clinical application, the entire this thing changes. You need to have a lot more of regulated stuff. That means you need to have approved cell lines. Is my voice clear? Okay. You need to have certified products from known people. You need to have quality control and quality assurance in every step. Okay, which we will be dealing with in your next semester when we talk about intellectual property rights and regulatory affairs. Okay, we'll talk about that more in that. But obviously, when we talk about, say, something like recombinant insulin or vaccine, you will not be taking it from any Tom, Dick, and Harry. You would definitely look for the brand. You would probably be more happy when you get it from a well-known brand or well-known manufacturer rather than from some unknown manufacturer. So it makes a lot of sense because all these people have gone through regulatory approvals. Okay. As I said, we will look at it from in the next this thing. So today, what I thought was we will continue our journey with the previous YouTube video where we talked, where the professor was talking about the ways proteins can be purified. We look at I think there is about 10 minutes or so that was left out. We'll see that, and then we will look at one way by which you could characterize. See, after you purify a protein, you need to characterize it. How do you characterize it? You need to have a functional assay for the protein. Okay. Typically, if you look at proteins, they could be receptors, they could be enzymes, they could be structural proteins, right? So if it's a receptor, you need to see whether the receptor that you have expressed, does it bind to its ligand, right? For example, every human cell, your keratinocytes, for example, they express a protein called epidermal growth factor receptor, okay, EGFR, epidermal growth factor receptor. So if you have suppose we express epidermal growth factor, you want to know whether the protein that you have purified, the receptor, is it functional or not, right? How do you know? By binding it with the epidermal growth factor, right? So as long as it binds to epidermal growth factor, you will know by various biochemical means whether the receptor has folded correctly, whether the receptor is able to act, bind to epidermal growth factor or not. Okay. It could be insulin receptor, right? So many different things. So these are called functional assays, okay? And functional assay basically tells you whether the protein that you have purified, is it functional or not. As I said, many a times when we purify our protein, uh, some of them may be dead on arrival. That means, you may always have a small fraction of protein that has lost its activity. Nevertheless, it gets purified along with the rest of the protein. Okay. So we have to be extremely careful. That is why when people talk about therapeutic applications, they need to have very high levels of characterization to know how much of it is pure protein, how much of it is active protein. Many different calculations go into that, which is beyond the scope of this thing. So we are not going to go into that. And let's continue our this thing on the purification and characterization. I may give a few these things comments in between the video, but is my screen getting shared? 1 minute. Huh? All right, here we go. Is it visible now?
Yes, sir.
Okay.
Very, very densely. And they're packed densely so I can get as many of these possible things in there as I can. Well, now instead of having charges
The
or holes or
URL has been
specific affinity
posted in the
molecules,
previous
now I
sync
basically
chat.
have a
So
bed,
I will
right,
post it along
that
with the
is
slides also, so don't
the
worry.
column material. I have a bed of hydrophobic side chains. What do you suppose is going to stick to it? Well, the things that are going to interact with those hydrophobic side chains are going to be hydrophobic molecules. And the things that are not going to interact with that support are going to be hydrophilic. So now I can separate on the basis of whether something likes water or doesn't like water. The ones that will come off of a column like this first are the hydrophilics because they don't interact with those C18 groups. The ones that are going to come off last will be those that are hydrophobic, that do interact with those. The rate with which they come off is actually a function of their hydrophobicity. So we could imagine a range of things that are very hydrophilic, very hydrophobic, and things somewhere in between. What I've just described to you, and by the way, there are a couple different strategies for HPLC, but what I've just described to you is the most common form. It's the only one you're responsible for. It's called reverse phase chromatography. Reverse phase. Okay? Okay. Now, I want to spend a few minutes telling you about a couple of techniques that now get into some really cool stuff with respect to purification of proteins. Okay? I'm going to skip down. I'm going to come back and talk about polyacrylamide gel electrophoresis later and SDS. What I want to talk about right now is an interesting technique called isoelectric focusing. Okay? Isoelectric focusing is a little difficult to conceptualize, but I'll try to do it here. Imagine, if you will, I now have a bunch of beads. And these beads have not one property, okay, but they're a mixture of beads, each with their own property. Right? So before I used all the beads that had the same hole, or they all had the same negative charge, or they all had the same affinity molecule, or they all had the same C18 group. Now I have mixtures of beads, each with their own property. What's the property? Well, the property is as follows. Some beads will have on them, let's say, 50 negative charges. And some beads will have on them, let's say, 49 negative charges. And some will have 48, 47, 46, 45. I go all the way down to 0, and then I have some beads that have +1 charge, and some that have +2, and some that have all the way up to +50, just as an example. Everybody envision that? So I've got some beads that have all these different things. So I take this slurry of all these beads and I shake them up, and I put them into a tube, a glass tube as I did before, and these beads are relatively mobile. That is, they can move around. They're not like the column I did before. Instead of standing it up like this, I lay it out like this. And now I apply an electrical current to it. What's going to happen? Well, to the positive end, the most negative charged ones are going to race and get over there. Right? And at the negative end, the ones that are the most positively charged are going to race and get over there. And right square in the middle, those that are 0 are going to stop right there. That make sense? Right? So what I've just made in this tube is a gradient of charge. A gradient of charge from the most positive at one end to the most negative at the other end with 0 in the middle. Everybody envision that? Okay, so this is called isoelectric focusing. It turns out that what I have just described to you in terms of separating charge also separates on the basis of pI. We talked about pI. pI is the pH at which a molecule has a net charge of 0. Okay? And so by setting up a column like this, I actually separate molecules on the basis of their pI, the pH at which they have a net charge of 0. The ones that have the lowest pIs will be at one end, the ones that have the highest pIs will be at the other end, and the ones that have the ones closest to a pI of 7 will be right in the middle. Everybody with me? Okay. Well, To do this kind of an experiment, to do this kind of a separation, I take not just the beads, but I take all my proteins and I mix it with the beads. I take all my proteins and I mix it with the beads. My proteins have a variety of charges on them. Some are very negative, some are very positive, and some are somewhere in between. When I apply the current, just as the beads separate themselves, so too do the proteins separate themselves. One end very low pi, one end very high pi, in the middle, those that have a pi around 7. So I've separated all of my proteins on the basis of their pi. Yes, sir?
Do you really need the beads?
Yeah, it's a good question. I do need the beads because the beads provide a support. In theory, I wouldn't need to do that, but if I don't have the beads there, the proteins just come racing off. So yes, I do need the beads there. Yes? No, it's a pH gradient, and because it's a pH gradient, that's where the PIs line up. So at a given pH, that's a good question, but at a given pH, okay, if the PI of this molecule is, let's say, 3.2, that means this molecule has a net charge of 0 right here, and that's why it migrates to that point and stops. Does that make sense? Actually, it's backwards of that, but yes. But you don't need to worry about that. All I want you to know at this point is that it is simply a separation on the basis of PI. Okay? Yes, sir.
So are the beads small like the powder? Are you trying to package many in there if you can?
Are the beads that small? No, the beads are not very small. The beads are relatively large. Yeah. Yes.
On the top picture, I don't understand there's the plus minus, and the minus. Is the three colors. Can you explain what?
Well, this is just simply saying that here, these guys are the most positive. They're going this direction. These are the most negative. This is going this direction, and the in-betweens are going to be in here. That's all that's saying. Okay, so keep it simple. Keep it simple. So we've got positive, negative, and basically neutral in the middle. I've got a gradient of that. Okay. So this is a way of separating proteins on the basis of pi. Now, this in itself is useful. For example, I say, well, my protein has a pi of about 3.2. I could go and cut out the band that correspond to 3.2, and I would have a mixture of proteins that all had similar pi to my protein, right? That's not the most important or the most powerful application of this technique. But in order for me to understand, for you to understand a more powerful application, we have to understand this process first. Okay? All right? So it's separating the bases of their pi. I've got a whole gradient of pi's. What's the next thing I do? Okay? Well, next time I'll tell you a little bit about gel separation, but I'm going to cheat and tell you about gel separation here. Okay? Now, keep in mind what I just told you about isoelectric focusing. We're going to use it in a second. But before we get to apply this technology into something else, we need to understand how we separate proteins. How many people have ever run a gel in a laboratory? Okay. Many people have. Gels are ways of separating molecules using electricity on the basis of their size. I'll talk about the theory for that in the next lecture, but today all we need to understand is that gel electrophoresis, as it's called, separates molecules on the basis of their size. The largest ones are the slowest moving, and the smallest ones are the fastest moving.
So this is pretty much what we see in DNA electrophoresis also, right? So that's the principle by which gel electrophoresis works. Doesn't matter whether it is DNA or RNA or protein. Okay, the smallest one will migrate the fastest, and the biggest ones will be the slowest ones. So they will be at the top, and the bottom will be the ones which have the least molecular weight. Okay.
Okay? It uses electricity to do it. As you might imagine, it involves charge. Okay? We'll talk about the specifics next time, but we're going to have gel electrophoresis separating proteins. So if I take my mixture of proteins and they've got a whole bunch of sizes and I apply them to the top of the gel, what will happen is the electricity will drive them through, the smallest ones moving the fastest, and the slowest ones, the biggest ones moving the slowest. Okay. Now, here's the clincher, and this is the cool thing. The cool thing is I can combine these two technologies. I do something called two-dimensional gel electrophoresis. It's schematically shown here. All right. The two dimensions are, I do two different techniques. First, I take my mixture of proteins, and I mix it with this slurry to do isoelectric focusing. So I take my tube, I lay it out here, I apply the current, I get the separation on the basis of PI. So I have this tube now that has this gradient of proteins separated on the basis of their PI, right? I'm very careful and I slice open this tube and I take that material that's in there and I put it on the top of a gel, and now I run electric current through the column material and driving those proteins into the gel. First I separated this dimension on the basis of PI. Now I'm going to separate all those guys on the basis of size.
So one quick this thing, you know. So this is called as isoelectric focusing. Okay. Now, as you can see, in when you do your first dimension, you're only seeing 4 protein bands, right? And when you do based on the second dimension, which is the based on the size, now you can see this resolves into 3 different proteins. This resolves into 3 different proteins. This resolves into 5 different proteins. This resolves into 4 different proteins. Okay. It is not that this is something that we do routinely. This first dimension one, we don't do it as routinely as this thing. This is generally useful when you're looking at a loaded cell, I say. Okay, when you want to characterize something that is totally unique, then probably you would be doing this. Okay, and for routine purposes, just doing this polyacrylamide, this thing should do fairly a good job, especially when we talk about recombinant proteins being expressed, as we also, whether it is PET or, any of those other systems, literally, 30 to 50% of the protein is going to be your protein. Okay, and the remaining proteins are going to be the host proteins. And you already would have done, a few steps of polishing. So you're going to have fairly 90% purity of your protein. So, it doesn't really need this. This is used only when you have complex mixtures. For example, when you want to know what is the difference between a normal cell and a cancer cell, you want to characterize it, then you would probably use this. What you would see is in a cancer cell, you may see once this protein may be either present or may be absent. Okay. So that tells you that that protein has some role for that particular cancer. Okay. Sometimes it may be present, sometimes it could be the absence of a protein. For example, if it is an oncogene, then it is supposed to be present. On the other hand, if it is a tumor suppressor, a loss of a tumor suppressor can drive the cancer, right? So it all depends on the which context. So we don't look for results and then we do. We actually look at the result and then we interpret the result. Okay.
Okay? What I will see is something that schematically looks like this. So if I were to look at this, okay, the molecules that had the most positive charge, okay, will be on the left side of this gel. The ones that have the most negative charge will be on the right side of this gel. And those that are the largest will be on the top, and those that are the smallest will be on the bottom. Down here, I would expect proteins would be small, positively charged. Over here, I would expect proteins would be large and negatively charged. Now, in 2 dimensions, I can separate every protein in this cell. Every protein that was in my mix, I can now separate and actually see a spot on this gel. Let me show you what this looks like. This I think is a magical technology, okay? This is what one might look like. Now, we see quite a bunch of interesting
I don't know when the thing got disconnected. Okay. We will look at another video which talks about characterizing. There are many ways by you could characterize. We will look at one of the technique for characterization. Okay. Is the screen visible?
Yes, yes.
Yes. Yes.
So
Hi
this
guys,
is a three-part series.
welcome to the last webinar, summer webinar from this season. The last webinar is called Protein Purification and Characterization. My name is Tania Pozo. I'm a member of the IGM Measurement Committee since 2018. And I'm currently a protein scientist in a company in California Bay Area. And today I will begin telling you a little bit more about protein biochemistry and fundamentals. As you know,
This is something that
as
we
you
all know,
proteins,
right?
they have different structures. You can have the primary protein structures where you have amino acids bound by the peptide bonds, but then these amino acids they begin to form hydrogen bonds, and they can form these two types of secondary structures that they are alpha helix and beta sheets, and there is also new types of interactions that these can bind together, like hydrophobic bonds or ionic bonds or van der Waals bonds, and then we get this tertiary structure where the protein is getting more the three-dimensional folding pattern, and then these protein these peptides can be form subunits. So then we get the finally quaternary protein structure, and this is how is the complete protein. And but why I'm mentioning this is because I really think it's important for you to understand that protein needs to be perfectly folded to get the function. And because of this structure of the 3D structure, we will get physicochemical properties for each protein. So now I will talk to you a little bit more protein solubility. So if we have a protein in a media, we need to provide the appropriate salt concentration in the media to prevent protein aggregation, and this is referred as salting in. And this means that if you have the protein, the ions of the salt, they will begin to bind around the protein, helping the protein to be soluble in water, and until a certain point where we will see here, as you can see here, where each protein is like a perfectly ionized with salts to be dissolved in water. But this is until a certain point, because if we add more salt, the salt concentration in the media will enhance protein aggregation, and this is called salting out, because in this case, if we have more molecules of salt, they will catch the water, and then the proteins, they will begin to clamp to each other, and then they will form aggregates. And sometimes this could be good if you want to concentrate protein, but it also could be bad, because then your 3D structure will get lost, and as you know, you can lose the tertiary structure or the secondary structure, and sometimes it could be reversible and your protein can get the right shape again, or sometimes it's irreversible, and that's bad, because then your molecule gets denatured. So proteins are ampholytes. What do you understand for ampholyte? Ampholyte contains two ionizable groups.
This is
One is
again something
acidic,
that you all know.
and the other
There's a
is
carboxy
basic.
terminal and an N
And
terminal,
proteins, they
amino
have this
terminal,
character.
right?
And they also have a R subgroup here that can change in each amino acid. So a medium's pH will determine the overall charge of the protein. What does mean that? So if we have here the protein at pH 11, sorry, not the protein, the amino acid will have a negative charge. And this negative charge at pH 11 will give you a net charge of -1. So in here you have the pH, and this pH will be changing. And then because of that, the net charge will also change in the amino acid. So at a specific pH level, the charge of these two groups can neutralize to create a net charge of 0. And this pH level is called isoelectric point. This is really important, because in this case, this amino acid at pH 7.6, you see, almost pH, almost 8, it will be 0. It will have, the charges will compensate. So you will have positive and a negative. And this is really important to know for each protein. So the pK is a constant that measures the potential for proton transfer. So it tells you how is the possibility of the protonation in each amino acid. So these pKs, they are constants that you can find in a table for each amino acid. And this is how we get to know the net charge of our amino acids, and then of our protein. And that's really important. So we will know what's the charge of our protein. So this is why I'm presenting you here a database called Expasy. It's the database that I like, but for sure there's many other databases that can do that. But in this database, you can compute the physicochemical parameters of a protein based on the amino acid sequence. So what you need to do is get the sequence of the amino acid sequence, the primary structure sequence of your protein, and then you need to paste it here in this space, and then you will press compute parameters.
So these days, as I tell, you have a lot of bioinformatics tools that are available. Okay. Previously, people had a tough time trying to understand, some of these properties of the protein. But nowadays, once you know the gene sequence, it's fairly clear what the amino acid sequence is going to be. And then, all you need to do is feed it into the XPASI database. It literally give you almost all the properties of the protein. You may still need to go back and confirm it in the lab, okay? But it gives you some ideas, how you
parameters,
could be doing
and
the
you will get a
experiment.
list of physicochemical parameters that you will obtain for each protein. And if you do mutations, like let's say that you had this protein, but you did many mutations, you could do random mutations, you could do site-directed mutagenesis, and you will have a new protein, and you need to do the same thing. You need to get the physicochemical parameters for each protein, even if you have mutated protein. So then you will get an output something like this. And as you see, there's a lot of information and you don't know what is what. So then I can tell you that here you are getting the molecular weight of your protein. So you know how big and the size of your protein. And this is good because this is good to know because in further purification steps, you need to know how heavy is your protein, how big, and then you need to know the extinction coefficient because this extinction coefficient is going to tell you how to quantify. You will use this value to quantify protein. You will also know the PI, where the protein is like a neutral out. So it's not positive, it's not negative, it's neutral. So and that's good. This is a value that you really need to know. And then you will also know how many amino acids you have with negatively charged or how many are they positively charged. And this is also good to know. So you will have an idea of your protein physicochemical parameters. So you can have a picture like this. So where you have two proteins, you have the human serum albumin and lysozyme. So as you can see, these proteins, they change the net charts at different pHs, like at pH 12, they are more negative. Sorry, so you see they are more positive. So you have to imagine that your protein in 3D is having different charges everywhere, right? And I want you to always imagine your protein in 3D. So, but why the protein net charge is important for purification? Because here in the middle is when your protein has the same amount of charges, positive and negative, and this is neutral. But if you see the pH, if the pH goes up, like say 7, 8, the negative charge, your protein will be more negatively charged. It doesn't mean that it's totally negatively charged, but it will have some positives, but the majority of the net charge, it will be negative. And here, when the pH is 5 or 2, or the protein, your protein will be in a state of positively charged. This is good to know because when you purify proteins, you will choose a resin, you will choose a column, and the resin will be full with beads, and the beads, they will have charges. So I will choose a negatively bead, a bead that is charged negatively. So my protein that is at this pH, let's say 5, will bind. And this is the way how I'm going to purify. This is called cation exchange, and this is called anion exchange, chromatography. So this is why it's really important to know the charges, the net charge. So reasons to purify proteins. So one reason to purify proteins will be the elucidation of a 3D structure, and you will use crystallography to do that. So for that, you will generate protein crystals, and this means that the crystals, you grow crystals. So you put protein that is really pure, highly concentrated, and then when you get the crystal, then you will shoot them with X-rays, so you will have a diffraction pattern, and this diffraction pattern will help you to build up a 3D structure. this is the real face of your protein. It's like you will see the face of the protein. And in this case, you will know if it's a dimer, if it's a trimer, or how is the conformation stage of your protein. But you also can identify where is the active site and then generate mutants in this active site and also get crystal structure of your mutants, so you can exactly understand how the substrate sits in the active site and which amino acids are interacting with your substrate, and this is how you will know the mechanism of reaction of your enzyme. You can also do functional studies, like characteristics of proteins or enzymes. Like for example, here I have a wild type from it's a luciferase from firefly. It's the one that produces luminescence in firefly.
I think the network is probably not very stable
it's
here.
a really well-studied protein. So there was a lot of scientists that they did random mutagenesis, and they find out that there was a enzyme that had enhanced activity. So this means that it used to bright longer. There was another group that discovered that they could also mutate the luciferase to get a thermostable mutant, because luciferase by nature is not a thermostable enzyme. So what we did is we put together these two mutations reported previously by other scientists, and we created the hybrid luciferase called YY5, and we measured the luminescence. So this is the luminescence. This means how intense was the light. And we use different substrate concentrations here. So we wanted to have little substrate, but really long light. And so here we compare all the mutants, and as you can see, YY5 was the best because using really little substrate, it could be brighter, produce brighter light. So this is a way to study functional characterization of wild types and mutants, but you can also study the physicochemical properties. So here I have the stability studies, where you can have the wild type here, and you can see that at 35 degrees, the wild type lost the activity of shining. It was not shining anymore, but the YY5 keeps shining for when it was 40 degrees, 360 minutes was still shining. So this is kind of like a good example to prove that your enzyme is supporting high temperatures. You can also do a study of how good the substrate, this is the luciferase substrate, sits in the active site. And for that, you do kinetic assays where you can see that you have a Km value. This Km value needs to be low, as low as you can, if the substrate sits perfectly in the active site. So the wild type had this value, but the activity mutant had lower. So this means that this mutant could accept the substrate better than the wild type. And our thermostable mutant was not good, not so good. But the YY5, the mutant that had the temperature and the activity mutations, it was in between. It was not as good as the wild type, but we had temperature in our, we could support temperature in our, and it was brighter light. So we were happy with that. Okay, so summary of this part, it will be, I will give you some tips as well, and it is crucial to understand the physicochemical properties of your protein of interest before proceeding to the purification steps. So evaluating the stability of your protein over a range of temperatures and pHs will allow you to optimize parameters for purification methods. Running stability assays will also indicate whether subjecting your protein to freeze storage condition is appropriate or not.
So freeze thaw is something that all of you must be aware, right? Especially in the current COVID situation. Many a times, these vaccines are shipped as one-time use. So an individual gets vaccinated and it's gone. But nowadays, it's like 10 people have to be there in that place, and then they will take out this thing and they will start injecting, right? So it has become different. But otherwise, many of these samples get frozen and thawed repeatedly, and sometimes in distant villages, when people go, you need to have these different thermostable versions so that they don't get denatured. Typically vaccines, they get denatured very quickly, so that is why you need to have a cold chain, but cold chain is not possible in every part of the world. So people are slowly working towards thermostable vaccine. That's what means stability studies means. Okay.
Purify the protein of interest according to the assays. The research that you need to run, for example, if you need to run crystallization, it will require that you have a high protein purity and high concentration. So that's the things that you need to take into account. And I think, Ian, we can go to questions if they have some.
So far, we have one question,
Okay.
and it was in respect to the XPASI database. Can this database give us the enzyme sequence of enzymes? And I think what they might be getting at there is the target sequence, the binding pocket.
Yes, there's other databases like Uniprot. We could add these links in the final, but there is a database like Uniprot and also NCBI. Like if you are looking for a specific sequence, but you don't know, let's say you want to find albumin, or then you can type albumin sequence, and then in Uniprot you will get all the albumins that they are officially in the database. So then you can copy that sequence and then use that in the XPASI. So then you can analyze your protein sequences and get the physicochemical parameters for your protein. Yes.
All right, that was the only question we had for that section. We will be taking questions at the end of the next one and the subsequent one after that. So.
All right. Okay. Any questions so far?
Yes, sir.
Yeah.
So, sir, in isoelectric focusing, the PI, the pH and the charge, they both are involved, right? It is not the different. They are not different process.
They are different process in the sense you will do first based on the charge, and the second dimension is based on the molecular weight. You will do them sequentially. So it's a two-step process.
Okay, sir. But in case of PI, that means the charge is involved.
Hi, is the separation for this thing. First step is based on the net charge on the protein. You saw one of the illustration in this thing. Every protein in your cell carries a total charge. Some have net negative charge, some have net positive charge. If you remember the previous professor, he said he will start with the bead that has 50 negative charges. Some will have 49 negative charges. Some will have 47. Like that he was telling. So you have this mixture that have various, charges on them, and then your protein interacts selectively to them. Anything that is neutral, which carries, both positive and negative charges on an equal, amount, they will be in the middle. Okay. Any protein that is rich in asparagine, glutamine, it's going to be positively charged. Anything that is, has extra, say, glutamic acid or aspartic acid, they're going to be negative in charge. So it is, and if you look at the database, your database, any protein will have a different amounts of these amino acids, right? There's no fixed ratio. So every protein is unique in that sense. It carries its own unique charge. So it is based on the charge you separate in the first dimension, and you based on the molecular weight you do it in the second dimension.
Okay, sir. Also, we have worked on UniProt. So, only by looking at the sequence can we find out whether the protein is modified or it is mutated.
Modified in what sense? Mutated you can definitely make out, because if a protein has a wild type and a mutant, it will already be available in the database. So you will be able to know whether it is a mutant or not. So that is not a problem. Modified, if you're talking about post-translational modification, then each post-translation modification has a certain motive, around which the post-translation happens. So you could in theory, you can tell, do I see any glycosylation motifs here? Do I see any other post-translation, motifs here? Those are all possible.
Okay, thank you, sir.
Okay, any other question? Okay, so so we will stop this part of gene cloning, which people commonly refer to as molecular cloning. Okay, so we have gone through an entire this thing, right? So we will stop this. We'll look at the cell-based cloning, cellular cloning from our next lecture. All right. So take care. Have a safe weekend.
Thank you, sir.
Yeah.